Hi guys, today we're going to be talking about photosynthesis. And I want to ask you to go and watch the Crash Course Biology video that I posted before you watch this video. It gives you a nice overview of photosynthesis and prepares you for some of the things I'm going to talk about. So if you started with this video, please pause here, go watch that video. There's a link on Converge, and then you can come back and watch this. The learning objective for you today is to be able to describe the purpose of photosynthesis and the difference between the light and dark cycle. So basically you are going to need to know the components of the light and dark cycle and how photosynthesis occurs. But overall, the idea behind this is once you know these, you should be able to describe the differences and the purpose. So this formula should look familiar. Basically, photosynthesis is cellular respiration in reverse. What you see is that plants use carbon dioxide and water to produce glucose and oxygen, which is great, this is why we need plants, because basically they take the CO2 that we breathe out and produce more sugar and more oxygen for us to breathe. So the equation has two processes, and really there's many stages to those processes. We have a series of light reactions. They are going to produce NADPH and ATP. And then we have a series of dark reactions where ATP and NADPH, produced in the previous ones, provide the energy and reducing power for a process called carbon fixation. So where is the site of photosynthesis? We have many different types of photosynthetic organisms. We have prokaryotes that are photosynthetic, and since they don't have membrane-bound organelles, this occurs in granules that are bonded to the plasma membrane. However, in eukaryotes, they have chloroplasts. And we have plants, but we also have algae and some other euglena that actually go through photosynthesis. So chloroplasts are interesting. They have a double membrane, a lot like mitochondria. They have an inner and outer membrane. But then they also have thylakoid discs, right? And this third set of membranes gets stacked in what's called grana. So these thylakoid discs are this third layer of membranes that are stacked in grana, where the trapping of light and production of oxygen, also called the light reactions, takes place on those thylakoid discs, whereas the dark reactions takes place in the stroma. And the stroma is just the inside between those thylakoid discs. So chloroplasts have this green hue, and that's because they have chlorophyll. And chlorophyll is going to be very important in the transport of electrons. It is going to be able to bind those excited electrons and transport them. And it's basically because it has a heme group. That structure is very similar to myoglobin, hemoglobin, and the chlorophylls that we saw in the electron transport chain. And it's this tetrapyral ring of polyphorons that you see over here on the left. So the reason that chlorophyll is green is actually because it absorbs light in the red and blue range. So basically what's happening is it's transmitting, or green light is bouncing back. There are some other accessory pigments in photosynthetic organisms that can absorb light at other wavelengths, but mostly they all are going to be chlorophylls. And those accessory pigments are going to transfer the energy to the chlorophylls. Chlorophylls are arranged in photosynthetic units. There are tons of antennae chlorophyll, and there's a lot fewer of these reaction center chlorophylls. So basically the antennae chlorophyll gather the light and then it's passed on to the chlorophyll that are at the reaction centers. And the chemical reaction of photosynthesis actually begins at these specialized chlorophylls that are at the reaction centers. So when my husband taught first and second grade, he taught a little bit of science, and one of the things he was teaching his students was that chlorophyll is a pigment. And there's a lot of different types of chlorophyll. And that's not quite right, but there are a lot of different types of pigments that are present in plant cells. So the whole idea is that chlorophyll is going to absorb light in the blue and red wavelengths. Okay, and because of that, that means what's left over is a green wavelength. So that's why we see plants as being green. However, in the winter, you start to change colors on your leaves and that sort of thing. And that's because other pigments are showing through, or what's happening really is the chlorophyll that is absorbing light at the red or the blue wavelength starts to go away. So you start to lose that chlorophyll, so you're going to see more transmittance of the red wavelengths. And that's an interesting concept to understand when it comes to visual light. I'm sure you guys talk about this at some point in physics, but the idea is that if a wavelength is absorbed, then you see the other wavelengths that are not absorbed. The light reactions include photosystem 1 and photosystem 2. Photosystem 1 is the reduction of NADP to NADPH, whereas photosystem 2 is the oxidation of water to oxygen. So you can see the half reactions and the overall formula. The reaction here is endergonic, meaning there is an input of energy. So where does this energy come from? It's actually coming from the light energy absorbed by the chlorophyll of the two photosystems. So this figure is really interesting because what it does is it represents electron flow from water that is going to NADPH. So basically what you see is that water releases electrons that are then excited at a wavelength of 680. And the energy relationship, I should point out, is shown on the left here as redox potential, with lower standard potentials at the top, and therefore greater energy as you go from the bottom to the top. And the thing is, energy is input as light. So basically what you see is that electrons are released from water, and then they're excited in photosystem 2. And then they're passed along via the cytochrome carriers, and then they're moved to photosystem 1, where they're excited by light again, which ultimately leads to the reduction of NADP, or nicotinamide adenine diphosphate, to nicotine adenine diphosphate with the hydrogens added. So basically, this is the flow of electrons. And the reason you're able to transfer those electrons is because they go from a higher energy state to a lower energy state all along the way. But how do they get the energy to get them to that higher energy state? They get it from light absorption. So basically, photons of light come in and excite the electron. So that's what we see by these two yellow arrows. Okay, you see another representation of photosynthesis. You see that electrons are removed from water and they're passed to photosystem 2. In photosystem 2, they're excited by light, and they're passed by a couple mobile electron carriers to cytochrome C. Cytochrome C acts as a hydrogen pump, similar to what we've seen in the electron transport chain. And hydrogen is pumping into the lumen of the thylakoid discs from the stroma of the chloroplasts. And then those electrons are transported to photosystem 2. They're again excited by light, and then those are transported in order to reduce NADP to NADPH. We also see that there's another ATP synthase that's used at the end of this. So that ATP synthase is going to use that hydrogen gradient that we learned about before with the electron transport chain in mitochondria to produce ATP. So the whole goal is not just to produce oxygen, it's actually to produce ATP for the plants. But in the process, it produces oxygen. So you can see we have all this moving of electrons, pumping of hydrogen, and ultimately that's going to be used to produce ATP. So what you'll see with photosystem 2 is that there is a system of different oxidation states from 0, which is the basal state, all the way up to S4. And basically what's happening is each electron is excited repeatedly. So 0 would be no electrons are excited, then electron 1, electron 2, electron 3, and electron 4, in order to release those 4 electrons from water and produce oxygen. As you can see, again, the reverse of cellular respiration. The net reaction of photosystems 1 and 2 is going to be the reduction of NADP to NADPH, and you can see that reaction here to the left. So photosystem 1 doesn't actually involve the splitting of water, and it doesn't involve the reduction of NADPH. It's basically independent, and it's harnessing this energy in the form of light energy that we call photons. So the photons are going to be used to excite electrons. And basically what it does is it pumps hydrogen into the thylakoid space, and then it gets dumped back into the stroma via this molecule that we've seen before, which is ATP synthase. So scientists have studied photosynthesis in a bunch of different organisms. One model organism that's been extensively studied is the Rhodopseudomonas. So this organism has a reaction center that contains a pair of bacterial chlorophyll molecules embedded in a protein complex that is, in turn, an integral part of the photosynthetic membrane. So basically what we see in this organism, which we assume is similar to a lot of organisms, is that there are these reaction centers. So there's a bunch of chlorophylls, but some of them are specialized reaction centers. And those chlorophylls, rather than just absorbing light, are actually going to be excited so that the light that's absorbed actually raises to a higher energy level. And this causes an excited electron that then passes along different energy levels. So this light absorption is going to occur at these specialized reaction centers. So I talked about accessory pigments before, but basically I want to point out they have specific positions close to the special pair of chlorophylls. So you have chlorophylls all over the place, and then there's these special pair of chlorophylls that are the ones that are going to be excited. The first of the accessory pigments is this pheophytin. Pheophytin is actually very similar to chlorophyll, and that's going to accept excited electrons, but the next electron acceptor is menaquinone. Menaquinone is very similar to coenzyme Q that we learned about before in the electron transport chain. The elastic electron acceptor is actually the coenzyme Q that we actually learned about. So you have a series of electron acceptors that are going to be passing the electrons from one to the next. So quinones are common electron acceptors in the electron transport chain as well as in photosynthesis. This is the structure of menaquinone and the structure of ubiquinone. So let's summarize what we see in the light reactions. So basically we can see that electrons are going to travel in the light reactions in a series of 3 different parts. Basically what we see first is that electrons are transferred from water to the reaction center of the chlorophyll at photosystem 2. And then these electrons are excited via light and then passed along an electron transport chain that's very similar to the mitochondrial electron transport chain. And then we see them passed on to photosystem 1. And what you'll note is that they're also excited here. So you're going to see an excitation of these electrons from light and they're ultimately going to be released and donated to NADP, which is going to be reduced to NADPH. So I talked about this before, but I just want to reiterate what couples the photosynthesis reactions to ATP production. So a proton gradient is generated across the thylakoid membrane, and this is what's going to lead to phosphorylation of ADP to make ATP. So this proton gradient is created by splitting water, which is going to release hydrogen ion, and that's going to be released into the thylakoid space. And then an electron transport system that we talked about with photosystem 1 and photosystem 2 is also going to lead to the pumping of hydrogen ions. And then when photosystem 1 reduces NADP to NADPH, it's going to use hydrogen ions. And ultimately, the flow of hydrogen ions back into the stroma through ATP synthase is going to provide the energy that's going to drive ATP synthesis or phosphorylation of ADP in order to make ATP. It's shown that ADP can actually be phosphorylated in the dark as long as there's a pH gradient. So if you look at this figure above my head, you see that scientists have taken chloroplasts at a pH of 4 and they put it in a buffer, and then they look at whether there is going to be a change in the level of ATP production. So how do they do that? They put it in a buffer with a different pH, which has a much lower hydrogen ion concentration, and then they add ADP and phosphates. And as long as the hydrogen ion concentration is changed, then they're able to make ATP. So the idea here is that this can occur without light. So that's the experiment that they're trying to show. This can occur without light as long as there's a hydrogen gradient that's occurring. So in summary, the mechanism of ATP production in chloroplasts actually closely resembles what's going on in mitochondria. The ATP synthase is very similar, and all the different structures are very similar. And just like mitochondria don't rely on light, the chloroplasts don't necessarily rely on light. They rely on the hydrogen ion gradient that's created. However, that hydrogen ion gradient is created by light. So we talked about the light reactions. Now we have to proceed to the dark reactions. And just like the video that we watched indicated, it doesn't mean that this only occurs in the dark. It just means that light is not necessary for these reactions to proceed, so they can proceed in the light and the dark. What you see is the dark reactions are a process of carbon fixation, and this is going to take place in the stroma. So what you see is that carbon dioxide plus NADPH and ATP are going to make glucose, and they're going to release NADP, NADPH, and inorganic phosphates. So this reaction has a lot of features in common with glycolysis and a few in common with the pentophosphate pathway that we didn't really talk about. But ultimately what you see is you're going to have carbon dioxide, and it's going to lead to the production of glucose. So this overall reaction is called the Calvin cycle, and it's named after Melvin Calvin, and he actually won the Nobel Prize in 1961 for chemistry. Very cool. So the Calvin cycle is really interesting. Basically, you're going to take carbon dioxide and use it to produce glyceraldehyde 3-phosphate. So every time carbon dioxide is donated to this cycle, and it's going to donate 3 molecules at a time, you're going to generate 1 molecule of glyceraldehyde 3-phosphate. So 3 carbons in carbon dioxide produces 3 carbons in glyceraldehyde 3-phosphate. However, let me ask you to pause here, because it's a little more complicated than that. Look at the way the carbons are incorporated. You have 1,5-bisphosphoglycerate that has 5 carbons, but there's 3 of them at a time that are going through. And it just keeps happening. You have to review this a few times. Now, the other reason I'm pausing here is I have an extra credit question on your homework. And so you get to replace a homework assignment just by answering this question. What is the molecule that catalyzes the addition of carbon dioxide to the Calvin cycle? So the Calvin cycle can actually be divided into 4 phases: preparation, reshuffling, isomerization, and phosphorylation. So preparation, basically you produce glyceraldehyde 3-phosphate and DHAP, which we've seen in glycolysis. Reshuffling, there's a bunch of reactions that are like the pentose phosphate pathway that we haven't talked about, that are just kind of reshuffling the carbons. Isomerization is going to be something similar that we've seen. We've seen the way that carbon-containing molecules can change the isomers. And then phosphorylation. Basically, you're going to have phosphates added in these pathways. So these are the reactions in the Calvin cycle. They're pretty involved, but the net reaction should be pretty clear here at the bottom. What you see is that 6 carbon dioxide molecules plus a bunch of electron carriers and water are going to produce glucose and going to release those electron carriers. So let's summarize what we have going on here. In the dark reactions of photosynthesis, which are also carbon fixation, what we have is that ribulose 1,5-bisphosphate is going to react with carbon dioxide, and that's going to produce 2 molecules of 3-phosphoglycerine. And we saw that in the reaction cycle that we saw. This reaction is catalyzed by the molecule rubisco, and again, extra credit, answer that question. But the rest of it's all about regeneration of ribulose 1,5-bisphosphate. All right, that's it for chapter 22. Let me remind you to go watch Crash Course Biology on photosynthesis and find the answer to that question about unintelligent design, because that's going to be worth some extra credit. Also, our last meeting via Zoom is on Friday, where we're going to review for the final exam. Again, the final exam review plus the answer key is posted on Converge, so be sure to complete that before we meet. And then our final exam is on Monday. It's not at a specific time. I'm going to give you 2 hours to complete it, and you can complete it anytime on Monday. All right, happy studying, scientists.